Hello, everyone, and welcome to today's webinar titled Ultra Fast One-Step RT-PCR Protocol for the Detection of SARS-CoV-2. This webinar is part of the Coronavirus Virtual Event Series, and I'm Susie Valdez of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, simply click on that Ask a Question box on the far left of your screen. As a reminder, the presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located on the top right corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Brian McNally, Chief Commercial Officer at Molecular Biology Systems BV. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Dr. McNally, welcome. You may now begin your presentation.
Good day. My name is Brian McNally. I'm Chief Commercial Officer at Molecular Biology Systems, and I'll be speaking today around the topic of ultra-fast RT-PCR for the detection of SARS-CoV-2 in the coronavirus webinar series. Molecular Biology Systems is a company that started in 2015 that's based in the Netherlands that has developed a next-generation PCR device. We'll talk about it, some of the studies that have been used around the world with the device. So as this audience is acutely aware of the pandemic that's going on, there are a number of headlines that have been occurring over the course of the summer that detail essentially how throughput of laboratory testing is impacting clinical value. Starting in sort of late July, Bill Gates came out with a statement that delays in coronavirus testing result in a complete waste of efforts, and they're insane. And I think this is probably best documented by 40% of U.S. COVID-19 tests are coming back too late to be clinically meaningful, based off of the fact that getting test results is essentially occurring too late in the window where the individual could then be infectious and then getting other people sick. And the backlog then is obviously making a headache for a number of efforts around the United States, around the world, where, for instance, Florida dropped a laboratory provider when a backlog of 75,000 tests was recognized. So there have been some papers that have come out challenging whether or not we should make compromises in laboratory testing around the difference between speed of results and sensitivity. In medRxiv, there was a paper released called Test Sensitivity is Secondary to Frequency and Turnaround Time for COVID-19 Surveillance. And they did some modeling in the paper where they contrast surveillance testing with true diagnostic clinical testing, if you will. And the authors concluded at the end of their publication that turnaround time and frequency should be prioritization. If you take the combination of these headlines and this sort of suggested strategy, that would suggest that testing rate, the actual rate of testing, is a key performance indicator for addressing testing within this pandemic. So what would be sort of the benefits that we would recognize from faster testing protocols? Well, first of all, obviously, faster testing leads to faster results, and then faster results, of course, can improve clinical decision-making, notably the decision when an individual should quarantine or not. Then, of course, increasing test capacity then per unit time allows any individual laboratory to then process more samples. If they can process additional samples, that could then result in more frequent testing of individuals. That has been recognized as something that would be needed for us to get back to work. And then, of course, if you can increase the number of tests that are accomplished per unit time, the associated labor and overhead costs that are associated with testing would be reduced on a per test basis. So if we have these benefits and we have these recommendations and these challenges within it, how can we go about doing this? And I thought the best way to introduce this concept would be to look at the total workflow as it is now. First, we have the collection of samples, and then we look at preparation, amplification, detection of signal, the analysis of the sample results, and then finally any sort of delivery of information to information systems. So if you look at this, then we have an opportunity to look at where are potential time savings that could be realized. And so we can see that what's happened so far this summer is there's been a real focus on collection and preparation. Notably in the collection, whether or not we can transfer from a health practitioner collection with a nasopharyngeal swab to something more like a mid-turbinate swab or an anterior swab that would allow for self-collection. To extend that, there's been a focus on saliva as a sample type due to its convenience and at-home testing possibilities or at-home collection possibilities. The next, though, is around preparation, and you can see that preparation for RNA isolation protocols typically takes 30 to 90 minutes. The 30 to 90 minute window for processing, for instance, 48 or 96 samples using automated robots. So in reaction to that, there have been a number of efforts by both companies as well as universities to develop solutions like lysis reagents and that would create crude lysates of viral RNA that would then be paired or used together with inhibitor-tolerant PCR chemistries or direct-to-PCR or RT-PCR chemistries. And both of these have resulted in time savings. But when we're doing an analysis of looking at what the long arm of this process are, amplification clearly stands out as one of the things that doesn't seem to have been greatly impacted in our response to pandemic testing. And so the question is, are there any methods that are available for decreasing this sort of 70 to 2.5 hour window of amplification? Well, there's certainly literature evidence that suggests that this can be done. There is additionally point-of-care device type methodologies that can do single sample acceleration down into sort of the 15-minute window, but those are poorly suited for dealing with thousands, if not millions, of samples that need to be tested. When we look at the literature, though, there have been publications, for instance, shown here on the left, where there was a three-step or a three-temperature PCR that underwent 30 cycles of amplification in less than 2 minutes. That technology was deployed by a group of Dutch researchers in a paper that was published earlier this year. The Journal of Microbiological Methods published a paper called Fast Identification of E. coli and Urinary Tract Infections using next-generation PCR. And what I think is notable about this publication is that what we can see is that their total workflow time, they listed as 52 minutes, but it included also DNA extraction, PCR, electrophoresis, then the staining and destaining of those gels, and then finally the visualization of that gel under UV light. So with that in mind, if we broke out just the number that they said for the cycling, it was a 16-minute protocol. So it suggests that there's methods available for accelerating thermocycling by next-generation PCR. So what is next-generation PCR? And we can start with looking at a history of thermocycling. Of course, the early PCR reactions used water baths, where 3 water baths were set up for denaturing, annealing, and extension of oligos and extension by DNA polymerase. And then a tube with a reaction mixture was then moved between these 3 water baths by a manual process. This process was then automated by putting it into a machine where then many reactions could be then put into a PCR plate, and this was all put into a Peltier block. And the thermocycle was then accomplished by heating and cooling that metal block that then passed heat into the well plates. So next generation PCR accelerates thermocycling using 2 mechanisms. And they're comprised by looking at the figures on the left-hand side, where we see first the lines that are diagonal between the resting, those would represent the ramp rates that occur during PCR cycling. And then, of course, there's the individual steps themselves that are highlighted in red, blue, and gray, and those would be the resting steps in each of the zones. If you can decrease to the point of elimination the ramp time, you can then result in a figure that's shown then there on the right. And that elimination of ramping rates for typical PCR protocols can result in a 40 to 60% time savings. If you can then further decrease the length of time that a reaction stays at each step, then you can also then decrease the protocol even further. This is accomplished by next generation PCR by two mechanical actions. The first is going to be then moving the plate between three temperature zones. And so I'm going to show you a video coming up where there's going to be a plate that's going to start on the left-hand side of the machine, and it's going to move into the machine proper, and there are going to be a pair of blocks. There's three pairs of blocks. The top are white and the bottom are yellow, and it's going to shift between them. The plate will be in red. As it goes through, each of these pairs of blocks, the white and yellow, are set to a specific temperature, either denaturation, annealing, or extension. The blocks will come together and compress the plate, and then subsequently compress the wells that are within each plate. And so I'm going to go ahead and start the video now. You're going to watch, again, the plate come in from the left and cycle through. And then at the completion of the video, I'm going to walk you through what the ramifications of compressing the plate are. So as we saw in the video, we can eliminate the ramp rates by using the action of moving the plate between three temperature zones. But I also referred to the compression of the plate by the blocks as another way to accelerate PCR. The plates themselves are a rigid frame that you can see here shown on the left-hand side that then have a ultra-thin plastic sheet that then is laid over top of them, where the wells are then formed into that sheet. The ultra-thin plastic is about 50 microns thick, and the wells are then laid out in a standard SBS format in either 96 or 384 well formats, and then can be various well volumes. After a reaction is then dispensed into the well, then there's a optical ready or an optically clear film that is then heat sealed on top of the plate before it's placed into the machine. With this in mind, then when those blocks compress, these wells actually deform, and the liquid inside has been shown to mix. And therefore, when the two very large thermal masses of the blocks compress this plate, squeeze the liquid thin, the transfer of heat is not just to the outside of the well, but it's throughout the entire reaction mixture, and it changes the temperature almost instantly. Therefore, there's not the need for these long dwell periods of, for instance, 30 seconds or a minute at particular steps, and we can draw those down to something that's more like 2 seconds, 5 seconds, 10 seconds in order to decrease the protocol further. We went ahead and paired that together then with and deployed that to a research group at the University of Bielefeld in Germany, led by Christian Karl-Schmidt, who then accumulated some results that were published earlier this year in medRxiv in a paper titled Ultrafast One-Step RT-PCR Protocol for the Detection of SARS-CoV-2. What you can see below the title there is a picture of one of those 96 well plates that then has been imaged on a fluorescent imaging system, like a gel documentation system that then clearly picked up a fluorescein signal. And then you can see the green and red outlines in those wells indicating positive and negative samples based off of the threshold. Overall, the paper was able to find a couple of key findings. They took WHO published assays and accelerated them in 96 well plates with protocols including the reverse transcription step that resulted in a 16-minute protocol. If you look at all three assays that were tested within the paper, they had a 96.6% sensitivity with remnant clinical samples that were used at 1/5 the recommended volume, suggesting a very sensitive detection method. Now, this methodology is a single target assay that highlights an effective screening protocol that could be used. And in the image that I showed here on the left, it's with one of the US CDC designed assays with an FDA cleared master mix that has been that was used in the paper. And so overall, the procedure looked something like this in the paper, where they selected the plates. In this case, they selected a 96 well plate that had a 20 microliter reaction volume. Of course, they come in different plate sizes and reaction numbers, but they're all SBS format and can be used with liquid handlers. They then use the optically clear films that I alluded to earlier to seal it in a heat sealer, using an anvil to then create a seal around each reaction chamber so there's no cross-contamination. After putting it into the thermocycler for 16 minutes, that accomplished the reverse transcription as well as the cycling, they took the plate, flipped it over, and put it onto a blue LED light imager for fluorescent detection. After capturing that image, a simple JPEG file, they then used a software package to analyze the fluorescent data and convert it into raw fluorescent units that were then able to be put into, for instance, an export of data analysis of a simple table. And so what you can see here is then a table that corresponds to the wells where their fluorescent values were captured for each of the different wells. This data then could be converted into an analysis software. You could look at threshold values. You could deliver it to, for instance, a laboratory information system. They continued this, and for instance, one of the programs that they accelerated was the Charité protocol that was published earlier this year with an assay targeting SARS-CoV-2 using the QuantiBio QScript XLT RT master mix. And they did a nice dilution series here with a synthetic RNA supplied by Twist Bioscience. And you can see in the figure on the left-hand side, there's sort of a decrease in amounts of copies per reaction, with the top row being the no template control representing the background of the reaction. And then above the plate, you can see the cycling conditions where there was an RT step followed by then a heat and activation of the RT reaction and then a 40 cycle protocol. And so as they went through this, they were then able to create a cutoff by taking the average fluorescent value across the no template control, adding 3 times the standard deviation to create a threshold value so they could call positive and negative samples. This same approach, then, the question becomes, well, what is the analytical sensitivity of a method like this? And in one of the examples, they've gone ahead and put together a figure that's shown here on the left, where they looked at the analytical sensitivity of the China CDC developed assays against ORF1AB as well as the N gene. And by using a dilution series and then seeing where it crossed the threshold value, you can see a really great analytical sensitivity of about 2.5 and about 4.5 copies per microliter. The same result, the same type of curves were also done for the US CDC assays, the Drost and Charité assays, as well as assays from the Institut Pasteur. So this kind of provides some support for using the assay as a sensitive methodology, but what would it look like, for instance, in practice? And so now, in collaboration with groups in Suriname and Molecular Biology Systems deployed 3 units there, 3 machines, and they've done a large study with clinical nasopharyngeal swabs that were collected and first tested with a Roche light cycler or a gene expert to identify their identities of the samples. And what you can see then is on the right-hand side a scatter plot that then compares the relative fluorescent units into assay from the US CDC on NEXTGENPCR as it relates to the CT value that was derived from the N assay on a Roche light cycler. And what I've drawn in the red lines there for you is the one that's crosses right around 58, 60 relative fluorescence units. Above that was considered positive by NEXTGENPCR and fluorescent detection. And then the vertical line represents the CT threshold cutoff that they're using for this assay of 38. And so that you can see then, if you analyze the positive percent agreement, there was 116 out of 121 samples were correctly identified, and there was 100% negative percent agreement over 350 samples, suggesting that this protocol is something that could be used with authentic samples. Now, when we think about this protocol in a little bit more detail, what we see is that these are singleplex assays. And most of the assays that have been used within the community are multiplex assays testing either multiple viral targets or the incorporation of process controls or human RNA controls. And so if we think about that, it really becomes a matter of looking at this detection bubble as whether or not there's some way to pair that together with the amplification in a way that allows for even higher throughput. So as I alluded, the previous studies all demonstrated singleplex readouts, and now the question is there a methodology that could then do the same with a multiplex readout? Of course, what I'm going to show you now is a process that then replaces that gel documentation system, that sort of blue LED light, and then works with a imager, a fluorescent plate reader. The fluorescent plate reader was then made available by BMG LabTech to laboratories in the Netherlands at the University of Nijmegen, and then, of course, in collaboration with NEXTGEN Life Science, performed the work there in the Netherlands. And what they used is the next generation PCR microplates are directly imaged. So they just take the plate out of the thermocycler and then put it into the BMG LabTech FlowStar. And they imaged the 96 well plates for both fluorescein as well as hex. The scan time for the whole plate took about 5 minutes. The well scan uses a 5x5 matrix, and they select the Grenier 96 F-bottom plate setting do the analysis. And now I'm going to show you some of the data that they did there using first a control experiment. They used a kit by Primer Design as a COVID-specific label with FAM. There is a process control that's included in that kit around using hex. And they started by creating a positive sample by pooling extracted RNA from 5 positive samples. The CT mix average was, the CT was about 20 with a standard deviation of about 1.5. And then they also used 3 negative samples as controls, as well as included controls for both positive and negative. And in all cases, they used samples of 8 microliters with 12 microliters of master mix. And when the sample was then run on a dilution series within the Quant Studio, you can see the CT values there on the right-hand side. So the dilution factor, there are 5-fold steps. So the first one that's shown there is essentially the sample's diluted 25 times, then 625 times, and so on and so forth. And the runtime for this experiment took 78 minutes. Using the same cycling protocol on next generation PCR, that protocol is shown on the left. That would take about 45 minutes compared to the 78 in the Quant Studio and 87 minutes in the Biorad CFX96. There is then the opportunity using that compression of wells in the next-gen PCR machine to then accelerate that protocol further. They developed a fast protocol in this study where you can see limited the denaturing time. They cut that in from 10 seconds to 5, and the annealing extension time from 30 seconds to 15, and maintained the 45 cycles. And so in very preliminary results, what you can see is the dilution series, and then what was the corresponding CT value for the Quant Studio in the table on the left. And then you see raw fluorescent units that were generated using the FlowStar for both the normal protocol as well as the fast protocol. And then those figures are then plotted in the histogram on the right-hand side, where you can see then the fluorescent values along the y-axis and the CT values along the x-axis. A negative control was then used as sort of a cutoff or signal, and what you can see is in an incredibly diluted sample that there's still sufficient fluorescent signal to differentiate it from a negative sample as well as a negative control. So when we take this study together, some of the key findings are that the fluorescent plate reader was clearly able to directly read the next generation PCR microplates. Next generation PCR successfully cycled a multiplex assay 3.5 times faster than the protocols that are currently in use. And there is a belief by the team that there's a further optimization of the cycling protocol could accelerate the assay even further. And then lastly, when you look at the dilution series and the wide range of fluorescent values, there's obviously significant dynamic range that then could deal with either very high viral loads or very low viral loads. And then in summary of what we've shown you today, when we think about the context of how much testing do we need, there have been estimates in the United States that we need somewhere between 200,000 and 1 million tests be performed per hour in order to successfully reach some of the estimates of what's required. And if we look historically, up to just a few days ago, the absolute peak rate of testing was only at about 49,000 50,000 samples per hour. So that's less than 25% of the stated goal by multiple organizations. And so if we think about that, and the impact of that, obviously the earlier statistics around the 40% of tests arrive too late to be clinically useful is a result of the fact that current systems can really only perform about 132 to 250 tests per hour. And so the results that we've shared with the community today are really about what next generation PCR system, a single unit system could perform. About 1,100 test results without pooling could be performed on a 384 well plate. They're estimates between 4 and 8 times faster than the current systems. The system, which actually is shown over my shoulder there, it only takes up about the size of 2 shoe boxes in total. And they use the exact same FDA-cleared assays, the same FDA-cleared RNA extraction methods. So it's an open platform that's available for development. So we're excited to share the studies and the progress of the team that we've been working with in both in Suriname, in the Netherlands with Isogen Life Sciences. So we're very excited to continue to work with the community, and we encourage people to contact us in relation to how they could use an ultra-fast RT-PCR protocol to accelerate testing results. So thanks. I wish you all very good health. And if there's any questions, I'll be logging in for the Q&A session later today. So thank you very much.
And thank you, Dr. McNally, for that informative presentation. And we will start now the Q&A portion of that webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. We have a few questions already coming in. Dr. McNally, since this is an endpoint cycler, can it be used for sequencing workflows?
Yeah, that's a great question. So the device itself can be used for sequencing workflows, gel electrophoresis, capillary electrophoresis, as well as this new sort of direct detection workflow. There's a number of organizations around the world that have already employed it, both in Sanger sequencing workflows as well as next generation sequencing workflows.
Thank you so much.
And just one additional point. One of the things that I described earlier on is that the plates are sealed with a heat seal, and to use them in the sequencing workflow, those seals are pierceable with a pipette tip. So you just remove the reaction mixture to then push it down the sequencing workflow.
Thank you so much. And can I run my own assays on the NEXTGENPCR?
Absolutely. The NEXTGENPCR platform is an open platform. As we showed in the presentation, there are a number of different assays that have already been put onto the machine. You saw the different ones that were published by the WHO for SARS-CoV-2, and then there have been a number of ones developed by private organizations that have also been run on the machine.
Thank you very much. We have time for one more question. Can the plates be used in liquid handlers?
Absolutely. The nice thing about when you're moving samples this fast and doing testing this fast is you're really going to need some sort of automation to be able to drive that workflow consistently. As we said, the plates are SBS format. They've been successfully deployed in both small pipetting stations as well as in large liquid handling platforms for both dispensation reaction setup as well as sample retrieval for sequencing workflows.
Dr. McNally, thank you again for your presentation today and for your important research. Would you like to provide any closing remarks for our audience members before we go?
Yeah, just a couple of things. We did thank a couple of the teams, and obviously I unfortunately left one out. The team at the University of Bielefeld has been a great supporter of ours and was one of the early adopters that we based a lot of this literature off of, so thanks to them. And then, of course, the NEXTGENPCR team is happy to work with organizations across the globe. So we'd be happy to be contacted either through the Q&A session or directly through our website at nextgenpcr.com. So I look forward to working with all of you so that we can improve the rate of testing. So thanks.
Dr. McNally, thank you again for your time today and for your important research. Before we go, I want to thank our audience members for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast will be viewed on demand for 6 months till March 2021. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed this live event. Until next time, take care, be safe, and stay healthy. Bye-bye.